All right, guys. Um, I'm not going to give you this answer, but if you can't figure it out, we'll talk about it when we get back from the break. Um, what would be needed to remove a peripheral protein from a membrane? So remember, a peripheral protein is only held to the membrane through electrostatic interactions. So anything that would disrupt the quaternary or tertiary structure of a protein would also disrupt the association of a peripheral protein with the membrane. So while a chemical reaction might do the trick, it's not necessary, right? But changes in pH, that would be fine. Okay, if you change the pH, then those electrostatic interactions are going to be disrupted. Phosphorylation would also be okay. Disruption of the entire membrane would also get it off, but it's not necessary, right? That would be for a transmembrane protein or another integral membrane protein. All right, let's talk about membrane fluidity. So I love this little GIF, which illustrates some of the movements. So this is showing the flexion of the phospholipid tails. And this flexion is going to aid the fluidity, aid the movement of the lipid molecules. Now, unless the lipid molecules are, unless their movement is restricted by something like a tight junction, which we'll talk about in a little bit, membrane lipids are free to diffuse throughout the cell, throughout the plasma membrane, or whatever membrane we're talking about. And this is very important. The diffusion of phospholipids, the lateral movement within that monolayer, they're only moving through a single monolayer, is important especially for the growth of the cell and the proper distribution of those lipids. Now, the flexion is going to also help the fluidity, as is the rotation. So you have this phospholipid, and it's literally spinning around its long axis. Okay, the warmer the cell gets, the higher the molecular movements, right? Just Brownian motion. So the higher the spinning, the more the flexion, and the higher the rate of diffusion. But the problem is, if the membrane gets too warm, it's going to get weaker. Okay, so you're going to allow the aqueous part from outside to go in and from inside to go out. Um, Now, we talked about the cytosolic monolayer and the non-cytosolic monolayer, and we said that they have different compositions, right? The outside of the cell is associated with carbohydrates, and the inside of the cell is associated with the cell cortex. Now, we haven't talked about membrane synthesis. Membranes are synthesized in the endoplasmic reticulum. And when you think about the internal membrane system, we'll talk more about this when we talk about the endomembrane system, but the non-cytosolic monolayer is always the non-cytosolic monolayer, and the cytosolic monolayer is always the cytosolic monolayer. But sometimes that gets confusing because in the ER or in the Golgi, the inside of those organelles, the lumen of those organelles, is the non-cytosolic monolayer. It's not facing the cytosol. So the cytosolic monolayer of membranes of the Golgi are the outside of those organelles. Okay, but when you get to the plasma membrane, when those vesicles fuse to the plasma membrane, then the cytosolic monolayer stays the cytosolic monolayer. Let me go back to this slide, right? Okay, so this is the outside of the cell, and the red is the extracellular matrix, and the orange is the cytosol, right? So if you're looking at this membrane here, this outer portion, of course, is the non-cytosolic monolayer, and this inner portion here is the cytosolic monolayer. But let's look at the Golgi, okay? Here, the cytosolic monolayer is on the outside. It's facing the cytosol. The non-cytosolic monolayer is on the inside of the Golgi, because the non-cytosolic portion of the Golgi is the inside, okay? So when this vesicle fuses to the plasma membrane, the non-cytosolic monolayer, which is the inside of this vesicle, becomes the non-cytosolic monolayer of the plasma membrane, which faces the outside of the cell. And the cytosolic monolayer of this vesicle, which faces the outside of the vesicle, becomes the cytosolic monolayer of the plasma membrane, which faces the inside of the cell. Yeah, I know that's confusing, but try to wrap your mind around that, okay? And that's really important because actually the chemistry of the extracellular fluid is very similar to the chemistry inside these endomembrane systems, right? If you think about the smooth endoplasmic reticulum, it's a repository for calcium. So is the extracellular matrix. There are very high concentrations of calcium out here. Whereas the cell spends a lot of energy pumping calcium out of the cytosol so that it can be used as a second messenger. Okay, so the pH of the inside of the endomembrane system is more similar to the pH of the extracellular matrix than it is to the cytosol. In addition, the carbohydrates. You have the carbohydrates being produced on the inside of the Golgi apparatus. And then those carbohydrates are they going to stick out into the extracellular matrix. Okay, let's go back down. Okay, so you have this asymmetry of the membranes, and this is maintained because flip-flop doesn't occur spontaneously very often. Okay, so flip-flop is when a membrane lipid goes from one monolayer and flops or flip-flops into the other monolayer. So here, non-cytosolic monolayer lipid is going to flip-flop into the cytosolic monolayer. That is a very rare event, unless there are enzymes. So in the ER, where you have lipids being synthesized, you actually have enzymes called flipase, and they're responsible for flip-flopping. You also have some flipases in the plasma membrane that are responsible for synthesizing certain phospholipids, but we're not going to talk about that. Okay, so flip-flop is very rare, though it does occur. Lateral shifts or lateral diffusion, movements of one lipid, phospholipid, through a single monolayer is very common and happens all the time. Okay, that's what keeps the membranes fluid, is the lateral diffusion within a single monolayer. So that happens very common and it's integral to the proper functioning of the membrane. Now, the fluidity of the membrane is really important because it allows the proper distribution of molecules through the cell. So it allows cell growth. It needs to be viscous enough that the inside of the cell doesn't go outside or vice versa. And it needs to be able to maintain some kind of ordered structure for the organization of the proteins as well as for mechanical support. but it also needs to allow molecules to move freely through the membrane. The cell is capable of changing the chemistry of the membrane, but it can't change the temperature outside. Hello, there's a big beetle in my room. Okay, I'm going to stop this right now and take care of this bug.